Hi everyone, welcome back to my A-Level Biology lecture series. In this lesson, we will explore the Mendelian patterns of inheritance. At the end of this lesson, you will be able to explain the term F1 and F2, interpret and construct genetic diagrams, including Punnett square, to explain and predict the results of monohybrid crosses and dihybrid crosses that involve dominance. Let's get started. Let us look at the Mendelian principle of inheritance. Genetics is a scientific study of inheritance which begins in the 19th century with the experiment of Gregor Mendel. He used the garden pea, the one that shows in the diagram, to investigate how traits or phenotypes are passed from one generation to the next generation. Mendel applied quantitative, basically involved counting, to analyze inheritance, making him the first to do so successfully. Although his findings were not appreciated during his lifetime, they were rediscovered in 19th century, at the end or the late of 19th century, forming the foundations of modern genetics. Mendel chose pea plants with clear contrasting traits, for example, yellow versus green seeds, and focused on the other 7 characters, which showing 2 distinguishable phenotypes. In this diagram, you can see that flower color with purple versus white, seed color yellow versus green, seed shape smooth versus wrinkled, pod color green and yellow, pod shape inflated versus pinched, stem height whether tall or short, flower position whether at the axial, mean at the side, or terminal at the top. So you can see that the bow phenotypes or the bow traits are dominant traits. In this lesson, we are going to focus on the seed color and also the shape of the seed. Mendel began his experiment by crossing through breeding. When we use the word through breeding, means homozygous. It can be either homozygous dominant or homozygous recessive. True breeding also means pure breeding. So Mendel started his experiments, by crossing the true breeding parental plants that show contrasting phenotypes for a particular trait. For example, yellow seed versus green seed. So this plant formed the parental generation, or known as P generation. When homozygous parental plant or true breeding parental plant with contrasting phenotypes for a particular trait were crossed, or the F1 or offspring in the first filial generation shows the dominant trait. In this case, when we cross yellow seed with green seed, you can see that all the F1 shows the dominant trait with yellow seed. So what is F1 generation? F1 generations refer to the first generation of offspring resulting from a cross between 2 pure breeding or true breeding parental plants with contrasting traits. So therefore, all individuals in the F1 generation are heterozygous, carrying 2 different alleles for the trait, and all of them shows or express the dominant phenotype. So when Gregor Mendel self-pollinated or crossed the individuals from the F1 generation, which are heterozygous, the resulting second filial or F2 generation show both dominant and recessive phenotype, typically in 3:1 phenotypic ratio. So the F2 generation or second filial generation refer to the offspring produced when the individuals from the F1 generation are crossed or self-pollinated. So in these diagrams, we can see that for the parental generation, there are two different or two contrasting phenotypes here, where we have the yellow versus green, and all F1 generations show the dominant traits, which are yellow color. However, the self-pollinations of F1 generation results in the F2 generation with yellow color seeds and green color seed in a typical ratio of 3:1. Gregor Mendel used the term dominant and recessive to describe the pattern in monohybrid crosses. Dominant basically means that the phenotypes that is expressed in all individuals of F1 generation when homozygous or true breeding parental plant with two contrasting traits are crossed. And the recessive means the phenotype that is masked in the F1 but reappear in the F2 generation in our example, which is the green seed. According to the Mendel law of dominance, when an individual or organism inherits two different alleles for a gene, in the heterozygous genotypes, the dominant allele is expressed in the phenotype. However, recessive allele is not, or the effect of the recessive allele is masked by the dominant allele. So dominant allele are denoted using capital letter, and recessive allele are denoted using lowercase letter. A heterozygous genotype results in the expressions of the dominant phenotype, or the effect of the dominant allele masks the effects of the recessive allele. Mendel also proposed two fundamental principles of inheritance. Let us look at the law of segregation. So each individual has two alleles for each gene, one from male parents, another one inherits from the female parent. During meiosis, these alleles segregate, so each gamete only receives one allele. So this process occurred during anaphase 1, where a homologous chromosome are separated and pulled to the opposite pole of the cells. So this means that a heterozygous individual, for example, capital Y and lowercase y, can produce two types of gametes, one carrying dominant allele, capital Y, and one carrying the recessive allele, which is a lowercase y. So during fertilization, gametes combine at random. So some offspring may inherit two recessive alleles, leading to the reappearance of the recessive phenotypes in the F2 generation. And this explains the 3:1 phenotypic ratio observed by Mendel in monohybrid crosses. The second law is the law of independent assortment. During gamete formation, allele pairs of different genes separate independently of one another. This means the inheritance of one gene is not influenced by the inheritance of another, provided that the genes are located on different chromosomes. So basically, we can get the gene, or we study the gene from chromosome 1 and the gene from chromosome 2. So how this allele orientates themselves during metaphase 1 is totally random and independent of each another. So in metaphase 1, the maternal and paternal chromosome line out independently. So each gamete can receive any combination of the maternal or paternal chromosome. For example, if capital Y and lowercase y are allele for one gene, and capital R and lowercase r are allele for another gene, and Y and R, these two genes are located on two different chromosomes. Then an individual with the genotype, heterozygous genotypes, Y and R, can produce 4 different types of gametes, which are dominant Y, dominant R, recessive Y, recessive R, dominant Y with recessive R, and recessive Y with dominant R. So this combination arises due to the independent assortment of allele at different gene loci. So this diagram summarizes the law of segregation as well as the law of independent assortment, which results in the 3:1 phenotypic ratio for monohybrid cross and 9:3:3:1 in the dihybrid cross. So at that time, Mendel's ideas were theoretical. The reason is because chromosome and meiosis had not yet been described. However, by the late 19th century, so scientists discovered mitosis and meiosis using improved microscopy. And these discoveries allowed researchers to relate chromosomal behavior to Mendel principles. So let us look at the monohybrid and dihybrid process done by Mendel. So a monohybrid cross involves the inheritance of single gene with two different alleles, where the two parent organisms differ in only one trait. So for example, we can always cross a yellow seed pea plant with green seed pea plant. So both of them refer to one single trait, which is the seed color. A dihybrid cross involves the simultaneous inheritance of two different genes, each controlling a distinct trait. So the parent organism differ in both traits. Example, we can always cross a yellow round seed plant with a green wrinkled seed plant. So in this case, we are studying two different traits, the seed color, yellow or green, and also the shape of the seed, whether the round or wrinkled seed. And we can always use Punnett square, okay, to study the inheritance. So a Punnett square is a simple genetic diagram used to predict the possible combination of alleles in the offspring resulting from a cross between two individuals. It shows how the allele from each parent can combine during fertilization. And also it helps to determine the probability of different genotype and phenotype appearing in the next generation, so either in the F1 or F2 generation. So in Mendel's pea plant experiment, capital Y is used to represent the dominant allele for yellow seed. Recessive allele for green seed is represented by lowercase y. Capital letter R indicates the dominant allele for round seed. And the recessive allele for wrinkled seed is represented by lowercase r. So let us study the Mendel law segregations where a monohybrid cross occurs in pea plants. When a true breeding homozygous yellow seed pea plant with the homozygous dominant Y was crossed with a true breeding green seed plant with a genotype of homozygous recessive Y, or the F1 offspring had yellow seed. So this is how the cross actually occurs. For parent 1, yellow seed, parent 2 with green seed. Then when we look at the parental genotype, because they are true breeding, so we use 2 capital letter Y to represent the genotype of parent 1 and 2 lowercase y to represent the genotypes of parent 2. So because yellow seeds parents have only capital Y or dominant allele, so therefore the gamete genotype will have all capital Y allele. For the green seed, the genotypes of the gamete is O with small letter or lowercase y. Always remember, when we write gamete genotype, make sure that we always circle the gamete genotype. So in this Punnett square, we can see that the F1 generation will be all heterozygous and shows the dominant trait, which is yellow seed. So a Mendel monohybrid cross begins with a parental P generation, where one parent is homozygous dominant for the trait, in this case yellow seed, and the other is homozygous recessive for the contrasting trait, in this case green seed. So each F1 offspring inherit one allele from each parent, resulting in the heterozygous genotype, capital Y, lowercase y. So since the dominant allele Y is expressed in the phenotype, so therefore all F1 individual display the dominant trait, which is yellow seed. Mendel then allowed the F1 heterozygous plant to self-pollinate, resulting in the F2 generation. So parent 1 and parent 2, in this case, both of them are yellow seed. And the genotype is heterozygous. And because of heterozygous, now they can produce two types of gametes. 50% of the gametes will have dominant allele Y, and another 50% of the gametes contain recessive allele Y. So the Punnett square here shows the possible combinations of the allele as well as the probability of the F2 generation. So we can see that we have 1/4 chance to get homozygous dominant, and we have 2/4 or 0.5 probability of heterozygous, and we can see that 25% of the homozygous recessive, which is the green seed. So F2 phenotypic ratio, you can see that yellow seed versus green seed. So we have 3 times of 0.25 or 1/4 and 1 time 1/4, resulting in this phenotypic ratio or typical phenotypic ratio of 3:1. So when two heterozygous F1 individuals are crossed in the monohybrid cross, the resulting F2 generation exhibit a characteristic 3:1 phenotypic ratio. And this predictable ratio supports the Mendel law segregation, which states that allele of the single gene separates during meiosis so that each gamete carry only one allele, and they combine when fertilization takes place. So at the fertilization, allele recombine at random, producing a mixture of homozygous dominant, heterozygous, as well as homozygous recessive genotypes. Next, let us look at the Mendel law of independent assortment, where we study a dihybrid cross in pea plants. When a true breeding homozygous yellow round seed pea plant was crossed with a true breeding green wrinkled seed plant, or the F1 offspring had yellow round seed. So this means that yellow is a dominant trait and round is a dominant trait as well. So we have parent 1 with yellow round seed and parent 2 pea plant with the wrinkled green seed. So both of them are homozygous plants, where yellow round seed is a homozygous dominant, green wrinkled seed with homozygous recessive. In terms of the gamete genotype, yellow round seed has all its gametes with dominant allele Y and dominant allele R. However, for green wrinkled seed, all its gametes contain recessive allele Y and recessive allele R. Let us look at the outcome from the fertilization. So all F1 must be heterozygous, and both or all of them shows the dominant trait, which is yellow round seed. In a typical Mendelian dihybrid cross, one parent is homozygous dominant for 2 traits, and the other is homozygous recessive. As a result of this cross, all F1 offspring inherit one dominant allele and one recessive allele for each gene, making them the heterozygous at both gene loci. So the F1 generation exhibits the dominant phenotypes for both traits, yellow and round. Next, Mendel then allowed the F1 heterozygous plant to self-pollinate, result in the F2 generation. So let us look at the parental plant 1 with yellow round seed, cross with the parental plant number 2 with yellow round seed. So both of them are heterozygous because they are F1 generation. So due to the independent assortment, their gamete contain either one of the following gamete genotype. 25% of the gametes will have dominant allele Y and dominant allele R. Another 25% with dominant allele Y but recessive allele R. Another 25% will have the recessive allele Y and dominant allele R. And the final 25% with recessive Y and recessive R allele. Let us look at the possible outcome when these gametes are fertilize each other. So from the cross of this F1 generation, so these are the F2 genotype as well as F2 phenotype. There are 9 out of 16 probability to show that the seed will be yellow round seed. 3 out of 16 of the seeds are yellow wrinkled seed, and another 3 out of 16 are green round seed, and the final 1 out of 16 will be green wrinkled seed. So F2 phenotypic ratio will show that yellow round seed to yellow wrinkled seed to green round seed to green wrinkled seed in a ratio of 9 to 3 to 3 to 1. When two F1 individuals are crossed, the F2 generation show characteristic 9 to 3 to 3 to 1 phenotypic ratio as predicted by Mendelian's probability. And this ratio provides strong evidence for Mendel's law of independent assortment, which states that allele of different genes segregates independently during meiosis, provided the genes are located on different chromosomes. And with that, we have come to the end of this lesson. Hope you have found this helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson and have a great day.